I think we're ready to start. It's a great pleasure to welcome Professor Simon Tavaré here as a Boeing Colloquium speaker. There's many places to mention. I even have my notes, but I promise it will be short. Professor Tavaré is a professor at the Department of Statistics and Biological Sciences at Columbia University, and he's also a director of the Irving Institute for Cancer Dynamics there. He obtained his PhD in probability and statistics from the University of Sheffield and held positions at Stanford, University of Utah, Colorado State, and University of Southern California. Before moving to Columbia, he spent 15 years at the University of Cambridge. Today, he will be talking about modeling of cancer evolution in single cells. Thank you.
Thank you very much indeed. Can you hear me okay? So I have an apology to make. I have two. The first one is that although your trees look beautiful, they kill me. So if I run out of air or start hacking, it will stop and I'll try and carry on. But this happened to me last week in New York, and they popped up outside my apartment and I spent a week off work. So anyway, we'll see how it goes. The other thing is that at my age, I can only do one thing at a time, and that hardly. I'm going to try and do two things at a time, which is we couldn't figure out how to get the presenter notes to do quite the right thing on this, so I'm going to try and use two machines. So bear with me if you wouldn't mind. So thank you very much indeed for the opportunity to give a talk. I'm going to talk about something we've been working on for maybe 3 years now, which is modeling and simulation of cancer evolution in single cells. To set the scene, let me just mention that the person who's doing the heavy lifting in this is Khan Din, who is now an associate research scientist in the Institute. And we've been working a long time with Ignacio Vasquez Garcia, who is I originally knew as a starting PhD student in Cambridge. And he's now a postdoc with me and with Saurabh Shah, primarily at Memorial Sloan Kettering. So he's a physicist by background. Khan is an applied mathematician by background. So, see, this is where I'm trying to do two things at once. It's all going to go pear-shaped, but we'll see. Okay, so I'm assuming that you're not all cancer experts, so I'm going to give a little bit of background about why we care about some aspects of the mutations and copy number alterations in tumors. I'm going to say something about what our IMAX-T project is about, which is how I got into this mess in the first place. So that'll be the sort of backgroundy bits. And then I'm going to talk about what the model is. So a bit of a discussion about statistical and stochastic models and clonal evolution. And then our main theme, which is how to model tumor evolution in single cells, a particular feature of tumor evolution in single cells, I should say. And then I'll say a little bit about what the inferential problems are and how we're trying to deal with them, and how we try and make the simulation produce data that looks like the real thing. And I'll explain why that isn't as obvious as it sounds. And then I'm going to say, if we have time and I don't run out of air, I'm going to talk about a control experiment from one of these single-cell DNA sequencing devices to show the rat hole I went down for far too long before I realized this wasn't going to go anywhere. And then I'll point out what the reality of these data is about and why I'm stuck. And then hopefully with a room full of smart people, you should be able to help. So I'm going to finish with hopefully 5 minutes left. Not a hope, but we'll see. All right, so let me start by saying a little bit about what this figure is about. So it has been known for a long time that various mutations are associated with particular cancer types. And this is you're not supposed to read this picture, but it's just to point out that across the top is a description of the name of the tumor type. And down the y-axis is the frequency with which certain mutations are found in samples from that tumor type. So I'll just point out a couple of things. If you have very sharp eyesight, you might be able to spot, I won't be able to spot, this is going to be hard, P53 sitting here. And in most of these, you'll find somewhere P53. I'm not going to try and look for them all because I'll fall over, but this is the plan. So lots of these mutations occur in many different cancer types. Some of them occur only in one or two types, and some of them occur with very high frequency and some with not such high frequency. So these things are assumed or are known to be something to do with the way the tumor evolves. And for that reason, they're called driver genes. They're the ones that we think are important. Not all of them, of course, are on this list, and we occasionally discover others. But it's probably fair to say that we've discovered most of the frequent driver genes in most common tumor types. That won't be true in two cases. One is rare tumors, and the other one is there's some problem with the fact that most of these data are collected from ethnically undiverse populations, mainly from Europeans or people of European ancestry. So it's not clear yet what the impact of that statement is. So these are mutations. They're positions in the DNA where something goes wrong at one or two base pairs. The other thing which is important in this, and this has been known for over 100 years, is that copy number alterations are common in tumors. So on the left is a karyotype of a typical, it's a metaphase spread from a standard cell. And what you see is the familiar mum, dad, one of each from each. What you see on the right is the karyotype from a tumor sample. And what you will observe, first of all, is that there's an extraordinary variation in the number of copies of chromosomes that you see. And also you can tell, this is a colored fish picture, you can see that various pieces have been translocated to different parts of different genomes or different chromosomes. So this feature is thought to be interesting, as that is, it's a driver of the mechanism of tumor formation in about half of tumor types. So roughly half are driven by mutations of the sort I just showed, and roughly half are driven by these peculiar aberrations. It's this thing that we're going to be interested in looking at. So to give you a taste of what happens, this is a picture of a more modern technology for looking at copy number variation in tumors. It's taken from a clone identified from a particular single-cell DNA sequencing experiment. And what's shown across the x-axis are the chromosomes, starting with the big one, number 1 on the right, on the left, and so on. And what is shown up the axis is the total copy number of that clone in that region. So these are measured to presage what's coming, these are measured by using this particular technology I'll say more about in a minute, in 500 kb, 500,000 base pair windows across the genome. So there's about 6,000 of them. So each of these is the average over a number of cells from this collection in these 500 kb windows. And what you see is the extraordinary variation in the copy number levels, the number of copies of each of the total chromosome count in a particular region from these things. It's really just a modern day version of the picture I just showed, but with no location information other than which area of a chromosome you're in. And what you can see if you stare at this is that often there's amplifications or changes in the structure across the whole chromosome, or perhaps in a chromosome arm. And I'm going to talk about both those aspects in a minute. So how did we get into this particular technology, and why do I care about this problem? So when I worked in England, we applied for what was called a Grand Challenge Prize. And we won the first of these in 2017. Cancer Research UK was very concerned that the way we were trying to identify and help cancer patients wasn't focusing enough on some of the key problems. So they suggested a number of things that we might focus on, we meaning our cancer field, and they promised £20 million, which at the time was worth more than it is now, I have to say, to try and figure out a response to some of these things. So the project we put in, and the one we got all the money for, was called IMAX-T, and the idea was to figure out how to image and annotate xenografts and tumors. So by annotate, I mean we want to image where the cells are in a biopsy, consider 100,000 cells, say. We want to work out where the cells are, and then we want to figure out 50,000 things about what's inside each cell in terms of its annotation. So, for example, what's being expressed, what proteins are doing what DNA you've got in there, and so on. So we set out on this thing some 6 years ago, and it's just now coming to an end. We promised originally to be able to do 4,000 breast cancers. I think we've done 4, And don't laugh, this problem turns out to be quite hard because we had to build a lot of the technology as we were going along. So I didn't build the technology, but I was involved in one bit of it. So what we do is to, maybe I can try and point at this screen. So we have a resected bit of a tumor or a PDX or a xenograft of some sort. And what we do is to embed this in a particular gel, and then we use a thing called an STPT, serial two-photon tomography device, to slice and image the little piece of tissue. What that imaging and slicing gives you is, first of all, the positions of the cells in the layers that you've got sequentially, so you know what's where to within statistical errors. And then it produces has a robotic device which collects the little slices and allows you in each of the slices to do some molecular biological technique in 2D essentially, because they're 10 microns thick only, it's the thickness of a cell. And what we do in those sections, we measure things, I'll say what in a second, and then eventually we want to put them all back together again to produce a 3D model of what the tumor is doing. So that was the project, or is the project. And the sort of measurements we would take would, for example, be measurements of mRNA levels, a technique which turns out to be really complicated to do in practice, but in principle tells you what's happening in this 2D sheet. We use imaging mass cytometry to learn about what proteins are doing. This is also three-dimensional. And then the bit that we have spent most of our time looking at is this little piece in the middle here, which is really looking at single cells, but without the spatial locations, but in which we can use single cell DNA sequencing, which is a bit more novel, to infer something about which regions of these samples might be interesting to look at. So we've been working a lot on the technology for doing the single cell DNA sequencing. Eventually, you have to put all this back together again, and you end up with this 3D picture at the bottom, which is telling you what's where and how they're annotated. Oh no, don't worry, don't worry. I'll carry on until I fall over. So it's, good God, what are you doing up there? What it is to be young. So anyway, we've not only built one of these single-cell DNA sequencing machines, we have one at the New York Genome Center, but we've been using it to do some serious science, which isn't the bit I'm really going to talk to you about. I'm just using it as motivation for why we want to look at this sort of data. So that's how we got into this. So this technique, I'm going to save time by not trying to explain all of these steps in this, but this is a tagmentation method, which allows you to segment and tag little fragments of DNA of length about 700 base pairs or so, plus or minus a few, tens or hundreds, and to then sequence those little fragments. And that technology we do in a multi-well format, in 1,000 well plates, basically. So we can do about 1,000 cells at a time. You can scale this down, make the pit smaller and do more cells. But the expense is, the cost of this is in getting enough information from each of the little wells. So this is shallow sequencing. We would typically only end up with something like a coverage of 0.1x, 0.1 of a chromosome across the, 0.1 of a genome across the whole thing. So there's very little information about mutations, but there's an enormous amount of information about copy numbers, it turns out. And that's the thing we're going to be modeling in just a minute. So this is now a well-tested technology. It's been revamped in many guises since it came out in about 2017 originally. And if you want to know more about the technology, Lax's paper in 2019 describes it. That was one of the group's papers. So what this sort of thing allows you to do is, first of all, we drop a cell into one of these wells, and while it's falling, we can figure out whether it's a cell which is, whether it's one cell in the droplet or two, or whether it's an S-phase cell, so it's one that's splitting into two. And then once we've figured that out, we can work out what's going on in each of the cells. And we can learn things like things about the morphology from the imaging at the top, or about where we are in the cell cycle, or about how chromosome missegregations work. We were already able to do things about mutations and structural variation, and one of the things that this is being used for is to find signatures that try to predict what sort of cancer you have. Are there particular features that are telling you what the tumor is doing? So we're adding things to the general picture of what can be done with this technology. So one of the things we're interested in is clonal substructure and the interplay with clonal selection. This used to be done, or is still done, in fact, with bulk DNA sequencing. This is find it and grind it for cancer biologists. So you take a bit of tissue, you mangle it up, you run it through a sequencer, and then you get historically, we found an awful lot of statistical trouble trying to reconstruct this mess of bits of genomes which are all scrambled up with each other. You don't know who's where and which came from which cell and so on. So hence the focus on single cell. Part of the problem here is distinguishing statistical and stochastic models, which I'll say a little bit about. And the statistical inference in this is still an unsolved problem. I've talked to several people about aspects of this today. So it's a common problem, not just in this field, but in every inferential field there is. So I'm going to focus on this copy number aware system. And in particular, I'm interested in what look like mechanistic models with interpretable parameters that describe the evolution of the tumor, at least in terms of basic features. The things I'm going to talk about are not spatial because we don't have enough information about how we would include the spatial nature of things. We can all write down models for it, but that doesn't mean anything in my world. So the other thing is that these things are usually, or look rather like, population genetics problems. So there's a long history of the sort of methods that are used to do this. The statistical approaches like dimension reduction and deep learning it depends on what the question you're trying to answer is, of course, but these are not necessarily biologically relevant or interpretable parameters, and so I'm not going to do anything of that sort. What we're interested in is a model for cell division, basically, and what we're trying to recapitulate are things like the little cartoon, the 3 pictures on the right, that show something about the modes of evolution. The classical one, which actually goes back to 1930, I think, described by Knoll in 1976 in a rather beautiful way, gives this famous-looking clonal evolution picture. You start with one clone at the beginning, and then a new mutation comes up, and that sweeps through the population, maybe doesn't get all the way, then another one comes up, and so on. Then there are papers around from actually some of my students, I must admit, got me into deep trouble here. They have various versions of this problem, which include the one, the so-called Big Bang model, which set off a bit of a storm of interesting discussion in the print about whether, in fact, these clones all start very early on in the history of the tumor. This is important to know if it were true. And then Williams's version, which has essentially neutral evolution. There's no inherent distinction between the rates at which the clones grow. And the truth is presumably some convex combination of all of these and a load of other things we haven't thought about, like what's in the microenvironment and so on. So anyway, what we're going to do is look at simulating the behavior of one of these tumor cell systems. So what we wanted to do was build a mathematical framework informed by driver mutations and copy number alterations and the effects of selection. For selection here, read cell division rate in some sense. So we expect some clones to evolve much faster than others or to replicate much faster. We need a simulation algorithm for this, which can simulate realistic numbers of cells millions of times quickly. So realistic numbers of cells means something of the order of 1 million at least. If we could do 1 billion, it would be better, but that's not feasible at the moment. We want to output realistic data, for example, where we can check whether our model is doing something roughly correct by looking at bulked sequences from the single cells, put them all together and try and infer what was happening. And I'll show you an example of that. We'd like to be able to say things about the structure of what these data look like, and I'll show you a horrifying statistical picture in a minute. And then we want a fitting routine to learn model parameters for a particular cancer type and data set. So the way this program is set up, is it's many, many thousands of lines of R and C and all sorts of other things, is that each tumor type has its own little sort of little file which gives the key information, but the rest of the structure of the thing is completely oblivious to what was happening. It just works from these little starter files. And then we want to use this framework to analyze aspects of what this technology, it's called DLP, is doing. And we were motivated to look at this because it turns out that one of the most famous techniques that's used to analyze this sort of data, it's been used to reconstruct the history of literally tens or hundreds of thousands of single cells now, produces very peculiar estimates of copy numbers when certain conditions are met. And it's obviously not doing something quite right. And we were trying to work out how we would test this. So maybe if we have time, say a bit about that. So what this model has to do is describe something about the turnover rate, what the cell's lifespans look like, something about the division probability, whether the cell divides or dies at the end of its lifespan, and that can be different for every cell, depending on the nature of its mutations and copy number and so on. We need to keep track of the copy number profile, so what the machines spit out is counts of DNA material or copy number depth at 500 kilobase intervals. So we want to be able to do that. And then we want to look at the driver mutation profile as well. That needs to be input because the cell's fitness depends on, and that then drives its division probability. So the parameters we need to be able to incorporate are selection rate parameters, copy number and driver mutation probabilities, and the population dynamics. So this simulation will generate any shape you want to have as the population dynamics, rapid growth, slow growth, whatever you like. And you can see a little picture of what one cell might look like in the middle. This thing here. This is the one that's divided up into these 5,000 little regions. And what you're looking at, the different colors refer to the copy number in that 500 base pair region. So they vary from 0, which is the darker blue. So this person is a female, there's no Y. To 1 is pale blue, 2 is gray, and so on. So 2 is the standard size, standard depth. And you can see some of the chromosomes are like that. But then you get some which have very high copy numbers. So when I put together several thousand cells of little images that look like that, you will go cross-eyed. But this is what we have to try and deal with. It's what we're trying to extract the information from. And we also add things about the driver mutation profile. So what's the role of TP53, which mutations do matter, which don't in this particular setting, and so on. So there's this sort of bit of mathematics behind this about what you might try to actually keep track of. And then we have to decide how things are going to divide. So there are lots of chromosomal methods, chromosomal mechanisms, which we can't say much about, but we can say some things about very important ones. One of the most important is whole genome duplication, which occurs commonly in tumor types and leads to essentially doubling the number of chromosomes across the whole genome. And these things are thought to be neutral. They can have very peculiar effects. They might be, for example, amplifying an oncogene and so on. So sometimes they don't do anything at all. We need to look at chromosome missegregation. So on the right are some images of what these look like in terms of the data in the bins, and they illustrate what these missegregations and chromosomal missegregations and so on look like. I think in the interest of time, which I've probably already lost, I won't try and describe every one of them, but let me just point to the things we want to include. So we want to look at chromosome arm missegregations. So that's when one chromosome arm is misplaced. It gets put in the wrong daughter cell, so you get an amplification in one arm. We want to look at focal amplifications and deletions. These are very common in tumors, and you could even see them in the little carrier-type picture I showed. And we would and so what we need to be able to do is to describe what the effects of these types of mutation are on the selective rate of the particular location that these occur in. So I'm happy to give anyone who wants a picture of these later, and you can actually have a look and see exactly what they all are, but I think in the interest of time I should swiftly move on. So let me say a little bit about what the selection model looks like. Again, I'm going to not go through all the gory details. So lots and lots of papers have pointed out that there's a close relationship between an arm's tendency to be gained or lost and whether that arm skews more towards tumor suppressor genes or oncogenes. So tumor suppressors are what they sound like. They suppress tumor evolution, and the oncogenes are the opposite. And so what this shows is for 3 particular types of mutation, what the impacts are on the fitness, which is the little circles with the colors at the top of each of the boxes. And this is part of what we have to model to describe how the process works. So this describes what happens when with a chromosome missegregation. So you see in the second 2 boxes, the top one has a copy number of 1, and the bottom one has a copy number of 3. So the 2 and 2 got missegregated to being 1 and 3. And that has led in the first example, because of what the selection rates were at the beginning, to an increased fitness for the top picture and a decreased fitness for the bottom one. And again, we do this as well for what happens for a whole genome duplication, what happens for an arm missegregation type of mutation. So in the simulation, of course, it produces a load of these, and you just have to work out what the effects of those are on the fitness of that particular cell. And then we have to combine them all into a model for how the whole population is evolving. So there are some of these features that we can't actually do very much about, especially very peculiar amplifications of genomes that occur in some tumor types, like chromothripsis, where you get completely mad-looking pictures with thousands of copies of things all over the place. We don't know enough of the mechanism to add that sort of feature. Fortunately, they're not that common.
Can I ask a question?
Yes, of course.
How are these fitnesses defined here? I should be able to read them out of the bar diagrams.
No, because they're probably too small. But let me go back and.
Yeah.
So, what the simulation has to do is describe what the selective rates are in each of these regions. So let's imagine that they're currently selection rate less than 1. Oop, I remember the trick here. It's okay. I shouldn't touch the screen. Selection rates less than 1, or selection rates are greater than 1. So there's scales so that 1 is the central point. And that shows at the bottom of the first box what they are now. And then what I'm showing is the effects of those mutation rates stay the same, and you've got to account for what happened as you went through all the mutations, and that will lead you to a new set of parameters for the next simulation of what that cell and its offspring do.
Okay, that makes sense. So that's selection with respect to some criteria about reproductive rate or something that's not directly
yes.
related to the chromosomes.
It is related to the copy number profile. So these are driven by the copy number profiles. There's a similar one that is this one that's driven by the effects of mutation. So for example, TP53, which is a tumor suppressor in normal conditions, that has a selective rate less than 1 here. That's the blue TP53. And then MYC is the other one, which is a notorious oncogene that has a selective rate bigger than 1. And what we base the selective procedure for the model for the driver genes and selection is how many copies of each of the different types there are. And again, I've illustrated here 4 different sorts of mutation, P53, chromosome missegregation, and so on. So again, the simulation produces these for every one it does. You produce a little table like this in silico, and it works out what the net effect of all those mutations is, determined by eventually the counts of the numbers of the original wild type and the mutant. And so it's fiddly, but I think you get the idea of what you have to do. It's rather unlike the usual sort of population genetics type selection model. So what you've just seen is the model for chromosome arms and driver genes. It turns out that most cancer types are driven mainly by large copy number aberrations, or many are, and some are driven by small events targeting driver genes, the 2 scenarios at the top. So you, of course, have a hybrid model which combines the 2 features in some sensible way. And I have not described how that's done, but it's mechanical. So in principle, we're equipped to describe a whole range of different cancer types using known information about the types of mutations and so on. So that's where we're headed. So, excuse me, the simulation algorithm, this is, I think, your bread and butter. It's basically simulating a Markov chain. It's not the dreaded Gillespie's algorithm, because that goes nowhere with a system like this. So it's based on some sort of careful analysis of tau leaping, which is a nice way to speed your way through the boring steps of Gillespie's algorithm, which don't move enough. So that has made a difference to what we do. It is, of course, approximate, and this problem is so complicated, I doubt we can prove anything deeply rigorous about what's happening, but that's what we try and do. And then we model the clonal evolution first, then we model sampling. So the idea here is to not have to simulate the entire tree of every cell and how they're all related in your million cells, because that will never go anywhere. So we do it by simulating clones first, and then we simulate copy number profiles based on those clones. We add some noise to make them look realistic. You'll see how in a minute. We then sample a number of cells, which is going to be our sample that we actually do the experiment on, as it were. So we get a sample phylogeny, and then we pour neutral variations of various types down that little phylogeny. So it's a sort of forwards, upwards, backwards, down. Yeah, I don't know how many times you go up and down, 2 of each, I think, to produce data of the right type. So this now will simulate millions of cells relatively quickly. So how do we actually do anything about the inference? Can somebody tell me how we're doing for time? Because I don't have a watch. Have we got about 20 minutes? 20, 25? Ah, thank you. 25, right, thanks. So I can even see that far. This is great. So what we're now interested in doing is trying to check some sort of boundary conditions about whether this actually looks like data. So we have a number of PCOG cohorts. This is one of the big sampling databases for tumors, which have been analyzed extensively for many different cancer types, but using whole genome sequencing in bulk. So not this sort of snazzy technology. So what we're interested in doing is fitting the frequency of gains and losses per chromosome. And we want to do it where there's a reasonable number of such gains or losses in the data. Otherwise, you're trying to simulate noise or infer noise. So the parameter that makes the difference is the ratio of the missegregation rate to the arm missegregation probabilities and the selection rate of chromosome arms. And what is shown on the right is the output from a particular ABC method called random forests ABC, which is, as far as I now know, the best and simplest way to do ABC on this type of problem. So ABC is about likelihood-free inference. As you can probably read off in a millisecond, there's no way in hell you're writing down a likelihood for this sort of problem I just showed you. So we simulate lots and lots of data sets from different parameter sets, and then we use this nice method of Raynal and Pudlow and others to infer the parameters one by one. There is no good way to do this in anything more than one at a time, unfortunately, in any field, not just this field. We're absolutely hopeless at doing 2 parameters and more. We may be good at doing 1,000 parameters, but that's going to be a bit tricky to impute anything from that. That can be done by other sorts of techniques. So anyway, we have a fitting routine for doing this. And what is shown are little posterior distributions of the get to exactly the right place. What you're seeing are the selection rates inferred from this model for particular chromosome arms. And what you should look at is the little line, which is so the pale blue is the prior and the dark blue is the posterior. And what you can see is there is at least information in the data. So it's concentrating the things. And what we use as our parameter estimate is, for example, the median or the mode of these things. Sometimes this doesn't work, as you can tell from this guy over here. Looks pretty hopeless. I'm not quite sure why that happens, but anyway, for the most part, it generates things which at least appear to give you a signal. I'm being very careful here what I say. So that's to do with fitting. Now, what you do is try and see what happens when you compare real data from PCOG with the parameters you've just estimated put into the simulation. So what you see are for 1, 2, 3, I think it's three different, I can't read, 3 different tumor types. So the one at the top is pancreatic endocrine neoplasms, and what you see on the top line is the data from the simulation, and what you see on the bottom is the data inferred from PCOG from this bulk sequencing. And what you see by and large matches up reasonably well. We've got the sort of amplification levels right and so on, and the selection levels right to produce data that looks like what you saw in the real thing. This, of course, is never the end point of anything, but it's just to try and calibrate whether the way we've included all these parameters is sensible. And it seems to be working okay. We can also, I'm going to skip that one. Let's see where I want to get to. Well, maybe I can. So we've then done the same sort of thing for fitting the selection model to chromosome arms. I won't describe the details, but we've also done this for what happens for fitting the model for driver genes as well. So can we say anything about the selection model? And again, it's the same principle. You find data sets where we know something about a tumor type that's not driven by copy number aberrations, and then try and infer something about the behavior of its driver genes, which are the thing that's driving that particular tumor type. And that seems to work well. Again, I won't bore you to death with the details, but there is a sort of standard recipe for trying to do this by matching up what we observe in the simulation with what we observe in the real world, and they match up pretty well in this picture. And that's illustrated actually in the thing on the bottom right. So now let me show you what the real data look like. Real DLP data. So this is a simulation. Well, this is a picture actually of a real roughly 1,000-cell experiment where the clones are these things we see with the 6,000 bins across the genome. And what you're looking at is the copy number. And the clones are inferred by some sort of clustering mechanism. And that picture is the clustered version of what these cells look like. So you see regions as you look along the chromosomes on the x-axis. You can see the different clones, and the different clones have different peculiar properties. For example, you see a region of high amplification right down here, and so on. And you can see a region of deletions, which have also occurred. This blue, I think I'm pointing in the right place, this blue line that goes all the way up here. So staring at these things gives you a headache, and B, it is not easy to interpret what you're seeing, because there are a lot of interacting things going on here. So what we want to know is, do our simulations look anything like this at all? How do we make them look like this? So let me show you what actually happens when you try and simulate this. So we actually know, of course it's a simulation, so we know the history of every cell in this. So what we do is add a feature to the story, which is we have this model which does all the selective parts. And then we add neutral mutations, which are copy number variations. These are actually well known in regular tissue now. They've been found in lots of different places and by lots of different groups. And we add also mutations, which don't change how things are evolving, but they will scramble up the picture you see when you do the clonal identification. So we can add a number of, at high rates, we can add a lot of this sort of background mutations, which are needed as well. And then we can start out with our simulated picture on the left, which you will agree looks nothing at all like a real DLP dataset, much too clean. And then we tweak it by adding these extra mutations. And what you see is a picture on the right. And it's getting a little bit more DLP-ish looking. But what you have to do in practice is also add a step which produces the errors involved in doing the sequencing experiment itself, getting from these pretty cells here scrambled up. We have to add another layer which puts in the mutations, or the bugs, if you like, that you get from the actual sequencing technology. So we have a routine, a separate thing which does that, which converts these, albeit now looking quite messy, but will look a lot messier when we add this sort of perturbations to it to make it look like the real data. So we think we've got a reasonable way of producing data that look like the real thing. So if you do that, you end up with this picture on the right, and this does look quite like the first picture I showed you, the real one from Salehi's paper. So this has got more of the features of a real DLP experiment. Again, clustered by anything you like, but UMAP is the one they typically use. So that's the end of this first little bit. I'm going to go through the second little bit, just a bit of. So we can describe cancer evolution reasonably well for many different types of copy number alteration. I've shown you, I think, 3 only. We can simulate large cell population numbers by some smart simulation technology. We have fitting routines which work at least decently well on single parameters so far. We need to do a lot more work to check it in boundary cases. For example, simulating populations to see whether you get roughly the right frequency of people with the tumor at certain ages and so on. And it can be expanded, and this is what we use. One of our applications is testing pipeline algorithms. So we would generate a whole load of data from DLP where we know the answer, and then compare the known underlying tree to the one that's inferred from the real DLP data and see how close we get. And that should lead us to better ways of thinking about the way the inference should be done for real cases, where they go through. I think the database now has not far short of 250,000 whole genomes done this way. So there's a lot of data. So part 2 I'm just going to skim over because it has an interesting toy probability problem in it, and it goes back to some very old things which I'm hoping some of you fires a spark and you can point out where I can find more about this sort of thing. So the way that DNA sequencing technologies work is pretty clumsy. I mean, it's amazing, but it's pretty clumsy. So you take a whole bunch of genomes and clone them, and then you chop them up, and then you try and reconstruct the order in which they came by sequencing these fragments. And you miss some, you get many copies of others and so on. But you get a picture which looks something like this one here. And from this, you are asked to infer what the sequence of the original thing was. So this is a standard technology. And it's the bulk sequencing method that's used in all of TCGA and ICGC. And in fact, essentially all cancer experiments are done this way. And it's now got very cheap to do. The hard part in this is not the technology, it's the getting the patient samples. So, here's what we thought we'd do. So we built, as I said, one of these machines, and we're interested in finding out how it works. So calibrating one of these things is not so easy. So we've done an experiment in which we run 50 wells in which you put a single cell of a cell line that you know is essentially diploid, so it's an H-tert cell line. And then in 50 wells we put 2 cells. And we know whether the 1 cells are really 1 cell or whether they're 2 cells in S phase. So we know what's 1 or 1.5 or 2, if you see what I mean. And then we produce the DLP data. And what we're trying to do here is mimic copy number variation of a whole genome duplication, because we've added 2 cells that are essentially the same, and that is going to mimic having 4 copies of each of the chromosomes, and that's a whole genome duplication. So from this data, can we tell whether we started with 1 cell or with 2? I have no idea, but I can show you what I've tried to do, and you can laugh later over a beer perhaps. So what's the observation? The observations are the depth of coverage of each base pair in the chromosome. So this is pretty exotic data. We have 10 to the 12th such measurements. It's a lot of data, and the data look like, wrong button, this picture. So along the top is the reference genome from 0 to G, and what you're looking at are 4 copies of a chromosome. This is a 2-cell example. And what you see in the little X's are where the shearing took place, where you fragmented the DNA. Again, this is the mathematician's version, you understand. And then the yellow symbols are the ones that are not sequenced, and the blue ones are the ones that are sequenced. And what we're watching is the pileup. So the pileup is counting the number at every base pair along the genome, which is 2.5 billion roughly, because of the regions you can't sequence, the number of blue things at every base pair. So it's called a pileup picture. And they can be computed relatively straightforwardly from sequencing data. So it's a very, very long string of low depth things, which should, of course, be no copies, 1 copy, 2 copies, or 3 or 4. But you do get some with 10 or 18. That's life. So let me show you what happens. So my toy model is the following thing. So if you had to think about this de novo, I spent 3 months doing the wrong thing or doing a different thing because I thought it was a really clever way of doing it. It turned out to be absolute crap. So I've tried a different strategy, which is much simpler, is also crap, but for a different reason. So I'm going to explain how. So the experiment has M haploid genomes. So when there's one cell, M is 2, and when there's 2 cells, M is 4. And remember, our cell line is essentially diploid, so that's what we should see. So left ends of fragments occur at the points of a Poisson process. Seems reasonable. We've fragmented at random. Independently in each haploid genome. Seems reasonable. And the fragments are sequenced with probability p each independently for every fragment. So that's about as simple a model as you can describe. So what you get from that process is a superposition of 4 Poisson processes. You've got the thinned one, and then you've put the 4 of them together. And so what you've got, this plot shows the overlaps. The sequence of depths in this Poisson model goes 2, 3, 4. If you follow the number of blue lines as I go across, 2, 3, 4, 3, 2, 1, 0, 1, 2, 3, 4, and so on. So you get data that looks like this overlap pattern. So it turns out, of course, that this is one of the world's best known stochastic processes. What's the behavior of the height process? What's the depth at any position in the genome? It turns out it's a very elementary Markov chain, which you can say everything, and I mean everything about. It's a birth-death process, it turns out, and it's a nice one. So it's something where I won't go through the details, but it's a birth-death process that pushes you back towards the middle of the state space. So if it's meant to be 0, 1, 2, 3, 4 for the counts, it pushes you towards m times p, basically. So this Markov chain you can say everything about, and what that allows you to do in this continuous version is to, for example, compute the probability of getting a 0 at position 2, whatever. So you can get the behavior of the whole process. In particular, its stationary distribution is binomial. This is obvious, of course. If you imagine looking at an enormously long genome and you're randomly picking bits with probability p, which is exactly what you're doing, then obviously the number of things you've got is binomial if they're independent choices. So this is exactly what you'd expect to see, and it is indeed what you see. So here are 2 examples. The first one is the counts of the number of base pairs in the real data which have coverage 0. So there's 2.63 billion of those. There's, I won't get the decimal places in the right place, but the next one looks like it's 193 million or something, etc. So that data here are the numbers of the counts. And what I did here was assume it was just we know it's actually a 1-cell experiment. So where on earth these 3s, 4s, and 5s came from, don't ask me. I'll explain in a second. So I just threw away all the ones that weren't 0s, 1s, and 2s and looked at the observed proportions and compared it to this little binomial. And it, I think when I saw this first, I thought, job over, Nature paper tomorrow. No. Not only because there's all those other measurements in there that shouldn't be there, 3, the 4, and the 5, and so on. I did it with a 2-cell experiment, so now there's meant to be 0 through 4, and that is what we saw. And you see the fit is also pretty good. Of course, trying to do something sensible with probabilities which are entirely inestimable because they're tiny is not going to get you anywhere, but the bulk of the distribution seems to be nice. And this is not this is not maximum likelihood anything. This is just equating the marginal distribution, the stationary measure, to the original observed data. So it's not really difficult statistical inference. So this is all very well. So I thought what I'd do is try and estimate using this Markov chain the parameters p and mu. Mu is the average, 1 over mu is the average length of a fragment, which we think is about 600 base pairs. And p is the chance with which we picked a fragment to be sequenced. So if you equi-space the points, then this process is trivial to analyze. It's classical inference for Markov chains from the 1950s, actually. My former boss in Sheffield wrote the original papers about this, a guy called Joe Garney. And so he would look at equi-spaced measurements across a time course like this and infer the parameters by maximum likelihood or any other method you want to use. And it's completely straightforward, apart from spending the odd 10-hour spell on Mathematica trying to figure out what the hell the eigenvectors of this thing look like. So there's some computational problems, but the idea is very simple. So what I thought I'd be able to do is compute the Bayesian information criterion that compared a 2-cell model with a 1-cell model with errors in it. So now they're all producing 0s, 1s, 2s, 3s, and 4s, but hopefully I could tell which one was a real 2 and which one was a real 1. No way. So it turns out that every time I tried this, I always, no matter which experiment I looked at, always picked 1 cell with errors rather than the truth that was 2 cells. So whenever it was a 2-cell one, it always said it was a 1-cell one with some bugs. So there's something not quite right with this. And so we started looking a bit more carefully about what's really going on with the data themselves. And I think I've got a minute to say something about this. So my toy model is clearly way too simple, and you maybe would have expected that. So let's not worry about that. So let me talk about this picture. I think this summarizes the problem. So we went back to the original data from the sequencing machine, and we looked at the aligned data. And then I decided that this looks like a model where you need to know the number of transitions from depth I to depth J in one step. So you would get an I to a J, and then you've got a long list of I's before the J at the end. And then you've got a long list of J's with the next J at the end, as it were. So, and we can count up the numbers of each of those. So this is the sort of information you need if you're looking at a hidden semi-Markov model of something. You need to know what's happening at the beginning and ends of all the intervals. So the data are shown in bold relief in the left-hand corner called N. And now this was a 1-cell experiment for which we had 1 million reads. So there's not a lot of sequencing. There's lots of zeros, as you would expect. So you go from 0 to 1 quite often. That is a spell where you've got no sequencing to a spell where there's sequencing depth 1. We went from 0 to 2 as well. There's some 0 regions that suddenly got 2. So where the hell they came from, who knows? Well, I have a guess in a minute. But then we go from 2 to 0. That's also peculiar, right? There's something odd in how the overlaps are being sorted out. The weirdest one is perhaps this little 9 in the middle here. Oops, oh, this is a good place to go. This 9, the red 9 in the top left-hand corner, is a very strange observation. It's the number of times you saw a span of length 1 followed by a span of depth 1. What? So what's going on? What is going on is some very unpleasant truths about the way in which sequence alignment gets done in these machines. And it seems it matters from the point of view of trying to do inference because the data are not quite what you think they are. So this region where, for example, there were 88 times where you saw a string of zeros followed by a 2 and a string of 2s followed by a 0, it looks like somehow 2 fragments have been lined up like this right next to each other. So when you read from here, you got 2, even though you hadn't found any before that. This is a peculiar sequencing bug. So what this experiment is aimed to do was to figure out what reagent depths, what reagent proportions we needed to get good sampling. at least in these data, we don't see anything bigger than 4. Before, we saw some things that were 18 and 20 and so on, which are obviously a mixture of not enough reagent in the wells, so maybe the wells are running out of, you're not chopping things up at random anymore, and so on. So there's a lot of features we have to try and figure out. And just to end, this is the killer. I have one more slide. So transitions from 0 to 1. So we're now looking at runs of zeros, which are ended by a sequencing depth 1 region. And you see the statistics of this. The median length is 2,000 base pairs. What? And the maximum one was 914,000. Don't really understand how you would get a span that long, given how this technology works, except I've now understood how. It was a boo-boo. This is including some of the unsequenceable list in the data, which was an accident, I think. More importantly, transitions from 1 to 0 tell you something about the sequencing experiment itself. So you get little runs of depth 1, which would typically be 1 chromosome segment, and then you stop, you haven't got any more information about that read, and you see totally different distribution than before, but something that looks much more sensible. This actually looks like what the TN5 transposase experiment is meant to do. It's chopping at roughly mean of 250 base pairs or so. Some of them are 800 long, but they're, sensible looking numbers, whereas the ones at the top are not. But then to confound life even more, I'll finish on this one. If you look at the distribution of the lengths of these singletons, so these are the lengths of 1s followed by 0, they are not exponential, of course. I mean, there's no surprise that this happened. There's something more like a sort of gamma-y looking thing. So all of those Poisson, the simple sort of Poisson process story is too naive for this problem. So now we've gone back to the drawing board to figure out why other errors in that little table are pointing to regions where we're doing the sequence alignment badly. And you have to get this right, because otherwise you're going to produce hundreds of thousands of cells with lots of flaky information in. So that's where we're at. Anyway, I will end with my collaborators. Skip that. So IMAX project I mentioned a bit about. Tan Dinh is Khan's father, who's a mathematician in Ho Chi Minh City, University of Sciences. Carol Naviciosu is my collaborator with esophageal adenocarcinoma. He's the guy who built the DLP machine that we use all the time, and the one who's been responsible for lots of this. And Sam Aparicio and Saurabh Shah were originally both at UBC and the British Columbia Cancer Agency, but now Saurabh is at MSK, and he's one of our closest collaborators. And Sam I've known for 25 years. We're sort of old pals. And Malhotra, Rhea, we have a lot of interns who come and work with MSK and us, and they're good fun to have around, and Rhea was one of the stars of this. Anyway, I have gabbled on too far. Thank you very much for your attention, and thank you for having me.
Thank you so much for the very interesting talk. We have time for maybe 1 or 2 questions. I don't know.
I'm curious, so when you were showing the bulk sequencing, one of the steps was first amplifying things, and so I'm curious if we thought about how it would change. I mean, having a toy model is easier when you only have 1 or 2 copies and everything, but do you think it's affecting the technology of the depths that you're getting? Like if you were to amplify, would you?
It shouldn't. I mean, that's what we were trying to find out. The question was, can we tell a 2-cell experiment from a 1-cell? So if we were given a whole genome duplication, would we know it was 1, or would we have no chance of finding out what we think? So it's trying to find the baseline for how you call copy numbers, basically.
But I guess I'm curious, if you amplified it first, would you find the depth is twice as big in one with 2 cells?
I'm not sure we can do, well, so this is an amplification-free technology, first of all. So that's part of the interesting bit about it. And it's also why you don't get huge coverage. I mean, apart from the money, but that's another reason. We've sort of gone back to the drawing board with the technology itself and trying to work out what amounts of reagent we need of all the different types that go into one of these experiments. This is where we've got to. This particular way of doing it, which is like 3 times the amount that is normally used, makes the experiments a bit more expensive, but it seems to give much cleaner data. But we're now down at the level of finding these other oddities that somehow we should be able to automate out of. And then maybe we can say something about what's really going on in this process, and maybe that would give us a statistic that allows us to distinguish ones from twos, because we would have the distributions would differ in some interesting way. And where I'm headed is some sort of horrible hidden semi-Markov model of some sort, where you spit out strings of things which correspond to these regions of ones or zeros or twos or whatever in some sort of coherent fashion. I'm hoping ChatGPT can give me the code, but I haven't tried yet.
One final question.
I guess you probably have a really interesting
I have a question. For this whole genome single cell sequencing, it seems it's very finicky. So are people who are doing that trying to, like, would different labs get very different results? Are they trying to?
No, so we're all friends. Let me restate that. Some of us are friends, but we all talk to each other. So there are, I think, 8 of these devices now. There's one in Cambridge which uses much bigger wells, which means you can get more sequencing done in the well. That gives you a higher depth of sequencing. There's one like that that Nick Navin has in MD Anderson. And then there are, I think, 6 or 7 of the original version, which is the one we built one of. There's one at MSK, there's 2 in Cambridge, there's 2 in British Columbia. So there's a bunch of them. And we have a working group which discusses both the analytical pipelines, which includes the New York Genome Center, who do a lot of this stuff, and another group that works on the sort of interpreting the sort of data you get, trying to make the algorithms a bit better. So we're pretty sure that most of these labs don't have time to do controlled experiments of any sort. There's one thing I didn't tell you, which is part of chromosome 20 in an H-tert line has a triploidy in it. And that's an interesting thing that we should be able to find. And we're trying to work on that too, but we need to get the data cleaned up first, or the algorithms rather than the data. And by the way, this is open. Anyone can join in this if you're interested in how these technologies work. They're always interesting. So.